Hi everyone, my name is Carrie McGrady and I'm a PGY1 acute care resident at Cape Fear Valley Medical Center in Fayetteville, North Carolina. I'm excited to be here today to present to you my project, the evaluation of traditional initial vancomycin dosing versus utilizing an electronic area under the curve to minimum inhibitory concentration dosing program. I have nothing to disclose for this program. Vancomycin is a glycopeptide antibiotic that has been in use since the late 1950s. Its target of efficacy is best measured by area under the curve to minimum inhibitory concentration, or AUC to MIC, as I will be referring to it as moving forward. However, this has been cumbersome to calculate, so we've been using surrogate trough concentrations of 15 to 20 milligrams per liter to represent this AUC to MIC target. However, the 2019 Infectious Disease Society of America guidelines now recommend an AUC to MIC target of 400 to 600 for both clinical efficacy and patient safety, as studies have shown that trough concentrations may overexpose patients and lead to higher rates of nephrotoxicity. Cape Fear Valley currently utilizes a dosing nomogram or first-order kinetic equations for vancomycin dosing. We rely on trough-based monitoring, and most of our vancomycin dosing is through pharmacy-to-dose consult service. We have approximately 450 consults per month. The purpose of this study was to compare differences in mean initial 24-hour vancomycin exposure using traditional trough-based dosing versus dosing recommended by an AUC to MIC dosing program. The primary objective for this study was to compare mean 24-hour vancomycin exposures. Secondary objectives including comparing mean predicted AUC to MIC and trough concentrations, comparing mean steady-state trough concentrations, and comparing rates of acute kidney injury, red man syndrome, allergic reaction, and hypotension. This study was a retrospective IRB-approved single-center study, and you can see my inclusion and exclusion criteria listed below. Patients for this study were identified via reports from Epic, our electronic medical record. Data was collected and kept in an encrypted Microsoft Access database. For the primary endpoint, each patient served as their own control, and for secondary endpoints, patients were grouped based on if the dose of vancomycin received matched AUC to MIC dosing recommended. And I will give you an example of that in the next slide. This is an example of how groups were decided. Patients in the trough-based groups were given doses that were different than the recommended AUC to MIC dose, which you can see represented here by the two red boxes. Patients in the AUC to MIC group were given doses that represented the recommended AUC to MIC based dose. AUC to MIC dosing recommendations were calculated using a validated online calculator. Trough-based dosing was calculated using either Cape Fear Valley's nomogram or first-order kinetic equations. Data were analyzed using students' t-tests and chi-squared analyses via JUMP 14 Pro. Here are the baseline characteristics for patients in this study. Most of the patients were male. The mean weight was 88.1 kilograms and the mean BMI was 29.9. The mean age was 55.7 years and mean serum creatinine was 1.1. The most common indication for vancomycin therapy was skin and soft tissue infections at 36.7%, followed by other indications at 23.9%, which included osteomyelitis and urinary tract infections. Then followed by sepsis at 26.1%, pneumonia at 19.7%, and finally bacteremia at 8.7%. Here you can see the primary endpoint results, differences in mean 24-hour vancomycin exposure. The difference was 268.9 milligrams between AUC to MIC-based dosing and trough-based dosing. This result was statistically significant. You can see that the AUC to MIC group had a mean 24-hour exposure of 2,380.7 milligrams, whereas the trough-based group had a higher mean 24-hour exposure of 2,649.6 milligrams. You can see the statistically significant confidence interval and p-value below. These are the results for the secondary endpoints. The results were statistically significant for estimated AUC to MIC and estimated trough level. However, none of the safety secondary outcomes were different between the two groups. For estimated AUC to MIC, you can see that the AUC to MIC base group had an AUC to MIC in the target range of 400 to 600, whereas the trough base group had an AUC to MIC estimated that was greater than the target range of 400 to 600. And this difference was again statistically significant, as you can see with the p-value and confidence interval to the right. For estimated trough, the difference again was statistically significant, but I would like to point out that the AUC to MIC estimated trough was only 13.5, whereas the trough-based group was 18.3. This difference should represent to us that AUC to MIC of 400 to 600 may not always represent that surrogate trough that we have been targeting previously. The results of AKI were similar between the two groups, with 10 patients in the AUC to MIC group and 11 patients in the trough-based group having an AKI. However, 17 of the 21 patients who developed AKI were receiving concomitant nephrotoxins as listed below, so that is something to consider. In conclusion, AUC to MIC recommended dosing resulted in lower 24-hour vancomycin exposures and also lower predicted AUC to MIC and trough concentrations. The results of this study support the new IDSA guideline recommendations, and it also may show a potential benefit in cost savings for the amount of vancomycin used for patients. However, this study also had its limitations. The majority of infections studied were skin and soft tissue infections, which are not covered in the 2019 IDSA guideline recommendations. These are non-invasive infections, and using the 400 to 600 AUC to MIC recommendation may be overexposing these patients to vancomycin. This also may have biased the difference in exposure due to targeting lower trough concentrations of 10 to 15 for skin and soft tissue infections. The study also had inadequate power to find differences in safety endpoints. I would like to acknowledge my co-investigators and the study site, Cape Fear Valley Medical Center, for allowing me to complete this project. Now let's wrap up with a self-assessment question. What side effect of vancomycin may be avoided by utilizing AUC to MIC-based dosing? Ototoxicity, nephrotoxicity, or red man syndrome? I'd like to thank everyone for listening today, and I'd be happy to answer any questions you all have about my project.